Well, good evening, everyone. Thank you again for joining me for another From the Clinic to the Living Room. Tonight, we're going to talk about vaccines. And given the climate of the last couple of years and also the cancer climate of things moving forward, this is going to be a really interesting topic for everyone. I hope that those of you that are joining us both on Facebook and on this webinar had a chance to take a look at the newsletter article that my special guest wrote this past month. It's really an interesting read. If you haven't read it already, that's basically going to be the topic of our conversation tonight. And so without further ado, let's get started. So my guest tonight is Dr. Kim Margolin from the St. John's City of Hope and Providence. She has so many accolades and has been, a real pioneer in the melanoma field for a few decades now. And I know that, you're part of the Cytokine Working Group and SWOG and ASCO. So who better to talk about all of the advances this year with the vaccines than you? So thank you so much for joining me tonight. Appreciate it so very much.
Thank you for inviting me. I'm looking forward to it.
Yeah. So I know that, most of our viewers are familiar with vaccines because, throughout their life, they've gotten vaccines as children, most likely. And especially now with COVID-19, vaccines are in mainstream everywhere you go. So, but just in case we don't really understand how vaccines work, would you mind taking a minute to explain kind of how a vaccine works in terms of, the general sense?
So in the general sense, if you put together if you sort of lump together all of the types of vaccines, of which there are many, and we may get into some of those details, the vaccine is some kind of material that is what we call immunogenic or antigenic, meaning it has the capability of being recognized by cells of our body's immune system, that is white blood cells, and those blood cells are born in the bone marrow generally, as well as in the intestine, and being recognized by white blood cells that are essentially taught by other white blood cells to recognize particles of those antigens and to respond or react against either those particles free-flowing or those particles as they are sort of attached or tethered to other cells. And then if we want to get more specific about how vaccines work, then we have to go into different types of vaccines.
Sure. And I mean, there definitely are multiple different kinds, attenuated vaccines, vaccine vectors, the mRNA vaccines, which actually in melanoma and in COVID-19 are extremely important, I know, moving forward. I know there's some DNA vectors, like some adenovirus vectors. But in general, so just to try to summarize, it's just a piece or a particle that is representative of a specific thing that your body then takes and makes an antigen against it, so it knows how to fight it. Is that roughly cool?
Yeah, and any of those vaccines that is made of either DNA, double-stranded DNA, as you were referring to, an adenovirus, or single-stranded mRNA, M standing for messenger RNA, both of those are what's called nucleic acids, and they're capable of encoding, that is creating a sort of a map of sequences of nucleic acids that encode for amino acids, which are what proteins are made of. And so that sort of roadmap gives rise to the proteins that are then broken up by special cells of the immune system and displayed on their surface to the cells that do the killing, which are generally called the T cells, or the cells that make the antibodies, which are the B cells. So there's certain things in common about all those immune reactions, and then there are differences depending on what kind of a reaction against the antigen is being evoked.
Okay, that makes sense. So when we are talking about, and you kind of touched on it briefly is when we're talking about the COVID-19 vaccines, there obviously are 2 mRNA vaccines, and then the Johnson & Johnson vaccine. What kind of vaccine is that? Is that more of like what we would consider to be like a traditional, what people would recognize as a vaccine?
Yeah, I don't actually know how many vaccines. That's an adenovirus vectored. In other words, the gene for parts of the coronavirus's genetic material is placed inside of an adenovirus and it's on the adenovirus's own chromosome. And when the virus gets into the body, it's handled, this chromosome is handled like the viral chromosome, but not only is the virus transcribed into RNA and then translated into protein, but also that piece of the coronavirus is also translated into protein. I believe that the J&J adenovirus is attenuated in some way so that it's not as pathogenic. It doesn't cause disease when it's going in, but then the piece that corresponds with coding for, say, the spike protein, the coronavirus, will end up producing a protein that looks very much like the spike protein. And that's what we want antibodies to. We want T cell responses and we want B cell responses. So antibodies as well as T cells that can kill cells that are infected by virus, by corona, as well the T cells help the B cells to make more antibody. It's a very complicated system, the immune system. It's got a lot of different players, a lot of different cell types, which I think is probably over what we want to discuss here.
Yeah, and it's definitely one of those things that makes it extremely complicated for patients when they're like thinking about, well, which vaccine should I get and which, what thing is right for me, and is this dangerous, and can this affect my cancer? And so I think there are because of the complex nature of the way that vaccines are designed, I think it makes it difficult sometimes for patients to know, that this is something that your body is trained to do, that is very normal, and vaccines have been around for a very long time. When we look at like preventative vaccines for cancer, I know your article talked about, a preventative vaccine for HPV, which is the human papillomavirus, and for both cervical and head and neck cancer, but also Merkel cell. I guess what is the basis? Is that like the start of how we have thought about using cancer vaccines for preventative measures? Like, where did that start? How does one start a vaccine trial for something like this?
So I think that over the years and even the decades, there's been a lot of parallel research in terms of cancer vaccines that are preventive or prophylactic against these, what we call the papovaviruses, the papilloma, polyoma, and variolated or something like that. So those vaccines or other vaccines, as others have been discovered in malignant cells, can be used in patients with advanced cancer. And that's kind of where most of this started. That was where the need was intense. And those vaccines in general have been ineffective, but have led to a lot of great research that has taught us a lot about those viruses and the role they play in cancer. But then they've also, some of that research or the resulting research has also given rise to the understanding of the role that's played by some of these viruses in oncogenesis, or the development of cancer. And that begot the use of the papillomavirus-based vaccines against cervical cancer and head and neck cancer, where you can actually, I'm not sure so much for head and neck, but for cervix, you can vaccinate individuals who are at risk for virus infection and thus for the secondary effect of causing cancer. And if there were a vaccine against hepatitis, it would be the same thing. If we could cause the infection to go away, that gives rise to a chronic immune reaction. So here you're seeing that sometimes the immune reaction can actually be damaging, and it can give rise to chronic inflammation. That in itself, in turn, contributes to the development of cancer. If we can vaccinate against that, then we never see the cancer, and all we have is a really good immune response against the virus that was going to cause the cancer. And even if you had to, to boost those, that's okay. I mean, those boosters don't have to be done every 6 months, as we think that might be the case for coronaviruses. It might be necessary only 5 or 10 years, like tetanus or some of these others, the pneumonia vaccine or things like that.
So the things that we have right now in order to prevent malignancies in prevention, just so that I can make sure that our viewers understand, are essentially malignancies that are linked loosely to like a viral infection. So you take a piece of the viral infection that then later, may contribute and allow the body to recognize that virus, thus one, making a memory cell, and two, making, the T cell that fights the virus in the first place, right?
Right. The memory cells are T cell subsets. And by the way, I think I made a mistake there. I didn't remember we do have a vaccine against hepatitis B. So we do have a way to lower the incidence of hepatitis-related hepatocellular cancer. And then it becomes a question of distribution, which we all know is an issue with respect to finding the people at highest risk of the disease where the virus is endemic, for example, like hepatitis B in Southeast Asia, for example.
Okay. So I think one of the kind of more complicated things, and I'm hoping that we can make it as basic as possible, is, how does the tumor hide from the immune system? So what makes the immune system not recognize cancer cells when it can recognize normal cells? So what makes that different?
Well, so that's the $64 billion question, and there are many answers to that. So if you want to use the word hiding and evading, you could, start by saying that there are probably structural components around tumors that might actually be physical barriers to T cells that might otherwise be capable of recognizing and killing cancer cells to just getting in. I think probably that's the least important of all the phenomena, because T cells are pretty flexible and they're pretty good at getting in. There are a lot of suppressive signals in the environment, in the microenvironment of the tumor, that include secreted factors, that include cells with the capability of suppressing T cell responses. They're capable of either of consuming some of the nutritional components that the T cells need. They're capable of creating an acidic microenvironment where the T cells don't work so well. There are regulatory T cells that are actually sort of telling the effector T cells not to do their job. There are there's a type 2 phenomenon, which can be one of everything: a macrophage, a T cell, a dendritic cell. And those type 2s are kind of anti-immune and pro-tumor, whereas what we want is type 1, which is pro-immune and anti-tumor. There are cytokines, which are molecules that signal between cells and allow cells to communicate with each other. And the type 2 cytokines are also suppressive against the effector T cells that could kill tumor. And the type 1 cytokines are, pro-immune and anti-tumor. So there's any number. This is why it's so hard to cure cancer with the immune system, because there are so many ways that the immune system can be prevented from killing cancer. And the other thing is the cancer cells can mutate. They can lose their ability to display the antigen on their surface, and so on and so forth.
So they can hide, they can lull the immune system into sleeping. So there's lots of things.
And they can change their clothing. Yeah.
Yeah, that's hard. So can we take, and this is like the $1 million question that we've been looking at, at our center over the couple decades, is can we actually take a patient's tumor and make a vaccine from it? And that's a question that we get asked all the time.
Yeah, of course. So that's probably the oldest method that's been used for trying to make effective cancer vaccines is to say, well, if the best source of the cancer antigens that would be unique to that tumor type as well as unique to that patient would be the ones that patient's tumor already has. And it could be one of many, many, many different kinds of antigens. There's quite a large catalog of types of antigens. And the fact is that there's never been a patient-specific cell-based or tumor-derived antigen or group of antigens that have been successful. And that's, to sort of summarize the reason for that is that probably all or many of the same mechanisms that the tumor has to evade the immune system when the tumor is in situ or in place are going to be exerted by this cancer vaccine as well, and cancer-derived vaccine. Yes, there are ways to make it more effective. You can treat it with different molecules. You can radiate. You can do weird things to it. You can encase it in things. You could wash the antigens off the surface. Many, many different things, but it hasn't been as effective as you might think. This is one of the reasons that now one of the most promising things that is being done is being pioneered by the same company, BioNTech, that is with Pfizer for the Pfizer COVID vaccine, which is to study antigens that are derived from mutations that cancer cells have as a result of damage from the environment, let's say. And the damage from the environment mutates the DNA, and then the DNA, as I said earlier, encodes or has the molecular map for RNA and then protein that is altered. It's not the normal protein, it's a mutated version of the protein. It may work the same way that the protein normally does, but because it's mutated, it may also be displayed on the surface of the cancer cell in this very special way that's required for the T cell to recognize it and then attack it. And so that's done with very, very sophisticated informatics where you sequence the entire strand of DNA from the patient's tumor. And then you use these computer algorithms to look for sequences, very short sequences, only 9 amino acids, nine little micro parts of the protein that are required for one part of the immune system or another, or 13 amino acids in the case of a different kind. And then you look at these catalogs, these computer catalogs of which sequences of 9 amino acids have been known to be antigenic in other settings. And then you create these vaccines out of maybe 10 of these or 20 of these. And then you put it in these lipid vesicles, and then you inject it just like we get our COVID vaccines.
Okay, so it would be almost like highlighting a piece of, taking a tumor and putting a highlight or a flag on it to make the immune system recognize it, essentially. Or
detecting a marker that's already there, and then making a whole lot of that stuff, of that mutant, and then using that mutant to do with the immune system. Yeah.
So I know that in melanoma specifically, because I know that probably a lot of the folks that are joined onto this webinar are interested in some of the mRNA vaccines that are being used right now in a melanoma setting. Can you speak about any of those in terms of how far they are down the road?
Yeah, I can try. I am not the world's expert in this by any means. The one from BioNTech has gone through phase 1s. I don't know exactly where they are vis-a-vis phase 2s, which would be the setting in which, having demonstrated safety and tolerability, you then build a trial that looks to gather up, let's say, 40 patients or so that have the same stage of the disease and approximately the same number of prior treatments and so forth, so that they all look as close to alike as possible. And then you test the drug and you see how the drug works. And then if it looks pretty good, then you do, of course, the randomized study and you compare it with some other standard. There are other companies that are doing something similar. And in fact, even though the concept of these patient-specific mutation-based antigen identification and then production of a vaccine is very appealing. There are still companies that feel that you don't really need to go to all of the trouble of sequencing the patient's entire DNA and trying to find those sequences that are antigenic, because there are still some what we call public antigens that are antigens that are expressed by many melanomas that are not expressed by any of the other tissues in the body, so that if you do a vaccine and you get T cells that are specific for cells that are displaying that vaccine, you're not going to get an attack on cells that would be dangerous to attack, like your eyes or your ears or your lungs or your kidneys or your liver. And so there are some vaccines that are made of these public antigens that are a little easier to come by.
Okay.
Yeah.
With that being said, if someone were to have gotten COVID vaccine where they've gotten mRNA and they were to want to participate in an mRNA driven trial, would that be something that would be okay? Would it be?
Yeah, I don't think just because they got mRNA. I mean, mRNA is very specifically coding for things. Sure. And I don't recall exactly how long the mRNA for the COVID, spike protein is, but it's going to be minuscule compared with something that comes from bits of the entire cancer cell's genome. So it's going to be very different.
Okay. So there would be no danger in doing those.
I don't think so. I mean, I could be wrong. I don't know what the company's requirements are, but it's hard for me to envision a company these days that would actually tell cancer patients that they can't be on a certain study because they got COVID vaccine that everybody should get.
Sure. Exactly. Okay. So I think one of the things that I wanted to make sure that I did was allow some time for questions. And we do have a couple of things in here, and some of these I'm not totally sure that we can speak to, but one anonymous attendee asked, which vaccine, and I'm assuming they're talking about the COVID-19 vaccine, would be considered to be the safest in terms of side effects or adverse events to the patient.
I believe, and my only knowledge of this is the same lay press that everybody in the audience is probably looking at, that all of the ones, I hope I'm not getting myself in trouble here, all of the manufacturers, at least the ones in the US and I guess Europe and Israel maybe, except for AZ, are equally safe. And that AZ is the one that's had very slightly more problems than the others, maybe J&J as well, but for the most part, they're all very safe. There are reports of myocarditis, and that's mainly the thing.
I think the biggest concern that I see in our practice are, the reports of, mRNA staying in the system for a long time and accumulating in tissues. And when you look at the science behind what an mRNA vaccine does, it doesn't make sense. It's metabolized and excreted just like every other protein your body is.
Well, yeah, the mRNA is not protein. The mRNA
No. I mean, like when you get like any other mRNA, like when you're creating mRNA, when you're creating proteins from mRNA for all the other cells in your body,
right?
That process is the same for your whole body. It's not special because it's COVID-19 mRNA is what I was trying to get at.
I don't think it would be special because it's COVID-19 specific mRNA. If it were encoding for a protein that for some reason bound very specifically to something that would be dangerous to bind to, then of course you could consider that dangerous. And I believe that that's actually felt, well, the cases of myocarditis and the cases of thromboembolic complications, I think, have been felt to be due to like autoimmune cross-reactivity between the spike protein responses, either the antibody responses or the cell immune responses, and some tissue. So that's my sense, especially in the case of platelets, where the thrombosis, the clots, have been due to platelet adherence and aggregation in vessels as a result of antibodies that are cross-reactive between spike protein or other viral proteins and the inside of the vascular structure, like a vein or an artery.
Okay. John also wants to know, or he asks, how would a vaccine that relies on a dysfunctional immune system of a cancer patient be advantageous over something like CAR T therapy, which manufactures the T cells outside of the body?
I'm sorry, I turned off the phone. Hold on a second. I'm so very sorry.
You're okay.
How could you repeat the question? Because I'm not sure I'm understanding quite what John wants to know.
So I guess he's asking, like, would it be more advantageous to do something like CAR T therapy where you're actually generating T cells in a laboratory outside the body versus trying to initiate a host response by giving a vaccine from a melanoma host?
The biggest problem with CAR T cells, besides all the manufacturing issues and the expense and the having to have the special lab, is that for solid tumors, there's a lot of problems with the local tumor microenvironment with T cells. As we talked about at the beginning of this question answer, the T cells may or may not have trouble getting into the precise location related to tumor that they need to. Mostly it has to do with the type of receptor that you engineer the T cell to have in a CAR T cell, or even in a T cell receptor transgenic T cell, which is somewhat different than a CAR. But what they recognize on the tumor has to be reachable, has to be within their ability to get to that cell and to have the antigen on a molecule that can interact with the T cell and trigger the T cell's machinery to transmit signals, and then have the signals from the T cell go outside and hit the surface of the target cell, which would be the tumor. And that has been very difficult to achieve with the technology for making CAR T cells, as well as the technology for making these T cell receptor transgenic cells that are similar but not identical to CARs. And there's a lot of research going on. And what I just said will probably be wrong in another week, because that's how fast the field is moving.
Yeah, I know vaccine and CAR T in general is just flying right now in terms of.
Right. Yeah.
So an anonymous attendee asked the question, do you recommend cancer patients receive the COVID booster shot in relationship to their treatment? I guess, are we recommending the booster shot? I feel like the answer is always yes in this
That's regard. The answer that I don't even let the patient finish their sentence before I say yes. And then I reassure them that these usually they're checkpoint blocking antibodies that they're on, PD-1, et cetera, that is not known and hasn't been shown to increase their immunity in such a way that it causes the vaccine to be more dangerous or causes more autoimmune side effects from the antibody. That unlike chemotherapy, it doesn't reduce their blood counts, and therefore you don't have to worry about getting it at a certain time of the cycle vis-a-vis the antibodies they're getting. And what is the other thing I tell them? Oh, that it's okay to get it with flu, although it's nice if you can separate it just so you know what they're reacting to if they do have a reaction.
And this is another thing that I feel like everybody's recommendation is different, but like in terms of, should they not get the vaccine the same day as the COVID vaccine, the same day as their immunotherapy, or should they separate it by a certain period of time?
I would never, I would never start the patient on a new drug the same day that they're getting a COVID vaccine. But other than that, I don't think there's any other precaution that you'd need to take about the timing.
Perfect. That's really helpful, because I think, patients are always asking questions,
sure.
How that is going. Another attendee has said, do we have any estimates of when an anti-melanoma vaccine would be FDA approved, as meaning, and obviously we can't determine that, but is it something that we think maybe is years away or decades away? I don't think it's decades.
Yeah. I think years is a reasonable sort of time unit. I don't think it's going to be months. I mean, I could guarantee you it's not going to be months, but it may be in the next short number of years. Yeah.
And I see more vaccines, at least initially, with melanoma, just based upon, the research that I've seen happen over the last couple of decades. I foresee it being more a preventative vaccine, maybe being a little more effective for melanoma than an actual metastatic disease without additional things involved, without say PD-1 or other agent.
Or it may turn out to be secondary prevention, which would be the patient in the adjuvant niche, where the patient has had a melanoma, they got adjuvant therapy, or their risk isn't high enough to get we're slicing and dicing that quite a lot these days, and it will be with PD-1 blockade, or it isn't, or there's some new molecule that it accompanies. So there's many different ways to skin that cat. Yeah.
Oh my gosh. And at the end of the day, I think vaccines in a melanoma setting would be absolutely wonderful to be able to use. So I don't want to take up too much of your time, and I know that there are some other additional questions coming in. I'll try to kind of filter those through. If there's anything that comes up, I'll forward along to you. But I appreciate you being able to take the time to come on and talk about your article. And I know that you do these in plain English articles for AIM at Melanoma pretty much every newsletter, or, especially when things come out new. So everybody that's watching, make sure that you check those out, because they're extremely helpful in navigating some of the really complicated science that happily is occurring right now in our field. So again, thank you again for joining us tonight.
Thank you for having me.
If there's anything else that we could do for you, please let us know. Have a wonderful evening.
Thank you. I see a lot of questions in the chat, so I'll see what happens with those.
We'll handle it later.
Okay.
Thank you so much for your help.
Okay. Bye-bye.
Bye.